So I thought as you're also a museum, I would focus on the work I've been doing with ancient DNA, essentially to get a sort of time series perspective. And sorry, let me just get this going. So quick overview of what I'm going to talk about. First of all, I'll just give a broad overview about the insights into selection that we can get from ancient DNA data. And then I'll look at two case studies. So one is going to be looking at some perspectives that we can get from ancient DNA data on positive selection in sticklebacks. And then the second case study is going to be looking at purifying selection in killer whales. And then finally, we'll come back for a summary slide. So actually part of a network of researchers looking at how we can use these ancient DNA samples to look at adaptive processes through time. And if you're interested in that, you can have a look at the website, which needs a little bit of updating, I should say. So it's www.adna.network. And obviously at the moment, it's quite difficult to do networking activities under the current conditions, but we hope to try and do some online courses in 2021. So maybe keep a lookout on the website and maybe the evilDare mailing list as well. And hopefully we will have some ancient DNA training courses going on online later on in 2021, as well as potentially other activities. And just some examples of the insights that we can get from ancient DNA. Can you see my cursor at all, Julia?
Yeah. Great.
So here we've got 3 examples of the same tree, and we see selection acting in population B at the same time. But we can see that if we just have present day data from populations A, B, and C, we might get different inferences, essentially from the allele frequencies. So we see the colouring of the branches here showing the change in allele frequencies. And we might see the dominant allele, the major allele in B and C being shared due to migration, for example. Or we might see that B reverts back to the ancestral allele due to migration from population A. Or we might see that B and C share a major allele, which is derived. And A, although A has the ancestral allele, it might look, for example, like it has the derived allele. And actually, there's a nice study on the FADS gene in humans, the insights that we can get from ancient DNA, looking at that example from Ian Mattison. That I think it was online as in a bioarchive, but I think it's now in a journal. So it's worth looking at that paper if you're interested in these patterns of selection. Now, the obvious signature that we can get looking through time is this change in allele frequency due to positive selection. So for example, if we sample a population at different times, time 1, 2, so that's just before an environmental change, and then 3 and 4 afterwards, we might see this rise in frequency of alleles that are associated with local adaptation. And just as like if we did a pairwise comparison between 2 modern populations, we might see differences in allele frequencies. Here we're actually seeing those changes in allele frequencies through time. And we've written a review paper looking actually at different types of selection. So not just, say, selection on loci of large effect, but also polygenic selection, purifying selection, and balancing selection. Just trying to give an overview of what the different signatures we would expect from time series data, but also a lot of the caveats that are involved with trying to get this kind of data from our ancient samples. Now, of course, what's actually more common than having a full time series of data is actually just having a single data point, a single ancient sample, just one individual, quite often maybe not even a whole genome. And even if you do develop later a much larger data set, which Ludovic Orlando has done with ancient horses, you might start off with one particularly, one sample and then build up from there. Now, back in 2013, this landmark study was done by Ludovic Orlando and his co-workers to sequence an ancient horse, which was actually over 600,000 years old. So it really pushed the boundaries of ancient DNA in terms of getting a genome from something that old. It's actually pushing the theoretical boundaries of just how far back we should be able to go with ancient DNA due to the fragmentation process and the length of DNA fragments that we need to be able to reliably map to a reference genome. So it's a huge achievement. And what did we gain from that? What would be the insights that you could only get from having an ancient horse genome that you couldn't get from modern horses? Well, all modern horses, even the Przewalski's wild horses, post-date the initial domestication processes. And so prior to the sequencing of this ancient horse, you would have to compare with an outgroup like the donkey. And then the genetic changes that you get by comparing a donkey with modern day domestic horses, they could have occurred all the way along this branch here. Potentially, you might also confound them with changes along the branch here. And by having this extra time point, you narrow down these genetic changes associated with the domestication process to this part of the tree here. So it's just a useful calibration point in looking at these evolutionary processes. So I'll start off with the first case study, which is looking at sticklebacks, free-spine sticklebacks. And we've got this nice little, I'm going to move, I don't know whether you probably don't see like the little box with Julia there, but she was covering this lovely illustration, which was done by Melissa Ngala, who has a website where she talks about her own research, but she actually does these really, really beautiful science illustrations. So if you're ever looking for a thesis cover, have a look at Melissa's website and you can commission her to do some work for you. And so free-spine stickleback are a classic study system in ecological speciation and adaptation. The marine form has many of these bony armor plates down the side, and that's thought to protect them from predators. And when they colonize freshwater, they actually lose a lot of these bony armor plates. And I think that's partly due to change in the predators. So they, for example, they might have less fish predators, but they might have more insect larvae predators. But it also gives them a growth advantage, so they can grow larger, quicker, and actually survive through the winters where the freshwater lakes or ponds might be frozen over up in the northern hemisphere. And we see the same changes in free-spine sticklebacks the world over. So this change is happening again and again and again. And there was a really classic population genomic study done by Felicity Jones and colleagues, where she sampled sticklebacks from around the world. We can see here, we've got the ones that are coloured, the dots that are coloured have got genome sequence and morphology, and the open markers are morphology. And freshwater and marine sticklebacks show a similar signature. And we can see here, for example, this is a Manhattan plot, which is showing the allele frequencies here. So it's FST or something equivalent to FST on the y-axis in the different positions. And we can see here the markers showing parallel changes in the genome. And it's this EDA locus, which is showing a large effect. And we know that that has a particularly large effect size on in terms of the phenotype changes in these bony armor plates. And there's many sites across the genome that show this parallel change in allele frequencies associated with this shift from the marine habitat to the freshwater habitat. Actually, there's a paper just gone online today where they've used experimental populations and they've released marine stickleback into freshwater ecosystems, and these changes actually occurred over a 12-year period in these experimental populations. So it can happen very, very rapidly. What we wanted to do was try and look at this through time over the longer time scale. Now, I should say those experiments that I just spoke about that have just been published today, they were done in the Pacific. And in the Pacific, a lot of these marine fish, they carry these freshwater alleles at low frequency. So we can see, for example, in this schematic, that this marine fish here has freshwater alleles. So it's essentially present as standing variation, and that's what allows it to quickly, rapidly rise to high frequency after this colonization of fresh water. Sorry. In the Atlantic, there's thought to have been a bit of a bottleneck where just a subset of Pacific individuals relocated and colonized the Atlantic. And so they just carry a subset of that original standing variation from the Pacific source population. So it could, in theory, be a bit more difficult, a bit more complicated for these stickleback in the Atlantic to colonize freshwater and go through such a rapid adaptation. And we wanted to actually look at natural populations going back in time, so hindcasting. And I decided a few years ago to try and look at these marine isolation basins where during the last glacial maximum, after the ice sheet started to retreat and the climate warmed, the land slowly bounces back. So first of all, these marine bays are exposed, and we can see that in the top figure here, the marine basin. And then just a little bit like when we get up in the morning, our mattress slowly bounces back up after we've got up out of bed. Well, the land also did that after the weight of the ice sheets was lifted and after they'd retreated. And so we slowly see the land start to rise above sea level. When that happens, first of all, these marine bays get cut off from the main marine body, so from the sea. So you get like basically a brackish pool where there's still a little bit of inflow from the sea at high tide. And then eventually, as the lake becomes higher above sea level, it gets completely cut off from the marine source water and becomes a freshwater lake. And we call these isolation basins or isolation lakes. And geologists actually use this change, which leaves a distinct sedimentary layer to calibrate the timing of the retreat of the ice sheets across like the coastline of Northern Europe and other parts of the world. And I've been working with geologists at NGU, so Anders Rommenset in particular, who's been using these cores to calibrate the timing of the change from glacial to post-glacial habitat in northern Norway. Now, occasionally, actually here we can see one of these cores here, so we can see this distinct sediment layer here. We can see like a brackish layer where we see a little bit of both the clay layer that's associated with the freshwater, and we see the bluish color of the marine layer here. And we can date, or I should say the geologists, they can date these layers by using organic material. So they would use pollen, grains, seeds, and other kinds of organic material to get dates of these different layers. And occasionally they find stickleback remains. So here we have bones from those layers that we were just looking at. In fact, from the brackish layer where the lake has just become isolated from the marine environment, but still is a little bit salty. And we can see here, for example, we've taken these bones here, and we can see that we've put them onto an X-ray crystallography image of a marine stickleback. So we can see that we have like bony armor plates and spines, etc. And the estimated dates of these bones from both of these lakes is roughly around the transition from the Pleistocene to the Holocene, so they're around 12,000 years old. And I also went up to Norway to actually collect some one stickleback, so it's kind of exciting. You have these reindeer just get you in the mood for Christmas. You have those guys just running around, and you have to kind of sneak past them to actually get these minnow traps into the water. So they're quite often around the lakes themselves. And I haven't got much reindeer handling experience, so I wasn't quite sure how dangerous they are, but no, I kind of came away unscathed, which is good. And we collected these freshwater samples and some marine samples from the area. Now, the two lakes are just 25 kilometers apart, and they're connected by the same marine fjord. And so all of these samples, there should have been the possibility of them coming from the same source population back at the end of the last glacial maximum. So first of all, we we did ancient DNA work on the ancient stickleback. We didn't get whole genomes, but we got in the case of one sample, we got megabases, and in the case of the other, we got like several hundred thousand base pairs of data, and this overlapped with both the regions that are associated with marine freshwater divergence and the regions that were not associated. We can call them neutral regions, but they're not. I think that's a little bit artificial to think of them as being completely neutral. Now, so we'll call them, I think we'll call them divergent and non-divergent regions for the rest of the talk. So the non-divergent regions, we can see here that the two ancient samples cluster with the Atlantic samples from that classic Jones et al. Paper. So again, we can see the distribution of the samples that Rusty Jones and colleague used in that 2012 paper. See quite a wide clustering of the of the Pacific samples, including one outlier down here. But along PC1, we see Atlantic and Pacific being separated. And I should say that the modern samples, when we just choose a single modern sample from Lake 1 and Lake 2, from our Norway site, or the fjord, they also cluster out here. So this isn't like an artifact that the two ancient samples cluster further out than the rest of the Atlantic samples. It just seems to be a bit of structuring within the Atlantic. And we can see also that the two ancient samples, when we're looking at the freshwater marine divergent regions, appear to be have marine genotypes. So we can imagine that they were probably marine phenotype, had a marine phenotype when they were alive. So had bony armor plates, for example. Now, when we look at structure, we see more or less the same thing. So we see that Lake 1 and Lake 2 form so here we're including multiple modern samples, and we see that Lake 1 and Lake 2 form quite distinct populations. Now, that's quite common for that to happen if you include multiple samples from one lake, from one population and another population, but then the rest you include in single individuals from multiple populations. And so we can imagine that Lake 1 sticklebacks, they would have shared a lot of their evolutionary history. There would be a lot of sharing of the drift, and frequent alleles that might be rare in the source population would be essentially rising to high frequency and be shared, and we would see this covariance of allele frequencies. So when we look at the PCA with multiple samples of Lake 1 and Lake 2, or we look at these sort of structure-like plots, in this case done with NGS adnex to deal with low coverage samples. And I should say also just using transversions to avoid any DNA damage, essentially spurious genotype calls for DNA damage. We essentially see the ancient samples looking a lot like the fjord samples and really looking a lot like the rest of the Atlantic samples, and just the two lakes looking like they have their own sort of peculiar ancestry, which is really just a subset of the ancestry that's out there in the fjord and in the Atlantic. And consistent with this, we see also the signature of a population bottleneck when we do PSMC. So PSMC is taking a single individual and it's looking at the patterns of coalescence across the genome. So essentially stretches of homozygous or heterozygous genotypes in these 100 base pair bins, which give an idea of how often there was inbreeding in the population, what the coalescent rate was, and through that, what the effective population size was. And we can see roughly around the time that we believe the lakes were colonized, this is around the time of our ancient sample here, we can see this this divergence from the marine population and this decline in effective population size, which takes place gradually over thousands of years in both Lake 1 and Lake 2. And we did this with multiple individuals, and we got the same pattern each time. We also looked at runs of homozygosity. So runs of homozygosity are essentially tracks of identity by descent, where you get identical haplotypes recombining in one individual, and they can be problematic. So they can actually be sources of deleterious recessive mutations that have been harbored in heterozygous genotypes and then exposed in homozygous genotypes. So they're quite often associated with inbreeding depression. And we can see actually quite extraordinary numbers of very long runs of homozygosity in especially Lake 2, but also in Lake 1, and very few in the fjord. Just to put this into context, the proportion of the genome that's covered in these runs of homozygosity in Lake 2 is roughly the equivalent to mountain gorillas or the Isle Royale wolves, which are a classic population for studying inbreeding effects. So it's very it's a very strong bottleneck that these populations have been through. And this is important for understanding adaptation and the strength of selection, because selection is dependent not just on the sort of the selection coefficients of the alleles that you're interested in, but also the effective population size, and that has a direct role on the efficacy of selection. And perhaps, not surprisingly, we see when we look at the ancient genome, and we're just looking here at regions that are associated with the present day fjord population or the present day lake population. And so they're at high frequency in one or the other. Now, we can see that the ancient sample mostly has regions that are showing a fjord-like signature, with roughly 25% showing a lake-like signature. So potentially, there's been some evolution in the population at the time that the ancient samples were alive already. But it's also important to remember that this might not necessarily be all due to selection. So here we're looking at essentially fixation in the fjord and close to fixation in the lake, and that could be due to drift in the lake populations rather than just selection on marine freshwater specific alleles. When we look at the regions of the genome that we know are associated with marine and freshwater divergence from the global data set in the Jones et al. Paper, again, we find roughly 25% of the ancient sample genome for what we have data for shows like a freshwater signature, and the rest shows a marine genotype. And what's interesting and perhaps surprising, especially since earlier on I was telling you about this study just published today, where within 12 years we have marine sticklebacks in the Pacific showing a freshwater genotype and phenotype. We can actually see that really it's only about 25% of the genome of the present day lake stickleback in these known marine freshwater divergent regions of the genome, which are actually showing a freshwater signature. Now, this is perhaps most surprising, is where actually some of these regions, like here on this sorry, if I press this twice, it goes onto the next slide, and it's got stuck. But this, I'll try and move it very carefully now. So this gene here, the GDF6, is associated with the loss of the pelvic spine. So that's typically a loci that's under selection in marine freshwater stickleback comparisons. And we can see actually that the freshwater, that the ancient sample had the freshwater alleles or freshwater haplotype at this locus, but actually that's been lost. So even though it was there in the ancestral population, selection doesn't seem to have been strong enough to drive it to high frequency or to fixation, and it's been lost where most of these present day stickleback have the marine genotype. What is interesting is that so we've arranged these by from most marine to most freshwater in the ancient sample. So they're not sort of based on the contiguity of where they would be in the genome. But we do see that a lot of the regions that were freshwater in the ancient genome are those that have become high frequency freshwater alleles in the present day lake population. But we also see that they're somehow leaking back to the marine populations as well. So these lakes now, due to this uplift, this post-glacial uplift, they're at the top of quite steep hills. It doesn't seem possible for marine sticklebacks to recolonize them. They're essentially little streams that come down from these lakes, but they're essentially waterfalls in places. So sticklebacks would have to swim like a salmon and leap quite significant distances to make it into these lakes. But it would be possible for a freshwater lake, for a freshwater stickleback to then migrate back into the marine environment. And that might explain a little bit why we see this high frequency of freshwater alleles in the fjord. It also could be due to conditions in the fjord, perhaps, like low salinity or something like this. So here at the bottom, this matrix here, we've got individual genotypes at these loci of particular interest where they're known loci of large effect. And for example, if we look at the EDA locus, which we talked about before, we can see actually it's quite pinkish. So it's got quite a marine signature in the present day lakes. But if we follow the path, we can see here in Lake 2, there's still a core freshwater haplotype central here. And we can look at that in a bit more detail here. And of course, EDA locus, we explained earlier, was that locus that is associated with this bony armor plates formation in marine stickleback and the loss of that bony armor plates, those bony armor plates in freshwater. So different alleles controlling for those different haplotypes. And I should say that our freshwater modern day lake populations, they were both of the low plated form, and the marine sticklebacks that we collected from the fjord were all high plated forms. So they fitted that typical phenotypic description of marine and freshwater sticklebacks. And here you can see Lake 1 and Lake 2, and you can see that core haplotype that we could see as blue on the genotype matrix earlier. But we can see that this core haplotype has risen to high frequency with different genetic backgrounds in Lake 1 and Lake 2. So for example, we can see freshwater regions rising to high frequency to the right of the core EDA locus in Lake 1, and to the left of the core haplotype in Lake 2. And it's just this core area that's highly differentiated. See the FST values here, highly differentiated in Lake 1 versus Altarfjord, which is the orange comparison, and highly differentiated in Lake 2 versus Altarfjord, which is the purple comparison. So essentially, our conclusion from this work is that the adaptation of stickleback, free-spine stickleback to freshwater, we kind of view it as this highly deterministic process based on the historic studies that have been done so far. And of course, it's a key study system for looking at ecological adaptation. But we find like an underappreciated role for stochasticity. And we're not the only ones to find this. There's been work by Juha Maria's group and others as well, where they find that lakes, especially around the Atlantic coastline, have not always shown such strong parallel evolution. And what do we think the reason for this is, based on our study where we've incorporated ancient DNA and modern DNA samples together? Well, I think it's a couple of things. I think first of all, it's this, you have a few different time points where you can actually get this stochasticity in the sampling. The first is the founder effect, where just a subset of individuals from the marine population colonized the freshwater system. Because of the way these isolation lakes form, it then reduces any gene flow, and especially immigration from the marine population into the freshwater population. So there isn't this constant influx of freshwater alleles afterwards. What you start off with is what you're stuck with. And obviously, then you're having to wait for de novo mutations to occur if the standing variation isn't there. And of course, what we saw with our ancient sample, in one case, was that a locus that had the freshwater alleles in the ancient sample still didn't rise to high frequency, so still wasn't present in the modern day freshwater lakes. And that was an insight that we could only get by having that ancient sample there, essentially from sampling that ancestral gene pool. And so that suggests that actually the efficacy of selection is weaker than is needed to drive all of those alleles that are present in that ancestral gene pool, but which are associated with freshwater adaptation, to drive those up to high frequency. So it's this combination of things, and it's this interplay between the efficacy of selection, effective population size, and selection coefficients. And I think it's a sort of an extra element of complexity to this free-spine stickleback system. And I think these ancient samples have got a good role to play. And the hope now, I'm just waiting to hear back from a grant decision in the next couple of weeks. And what we aim to do is actually go out and, rather than just opportunistically wait for the geologists to find these sticklebacks in the sediment cores, is to go back and target some of these lakes, drill multiple cores, and see if we can actually get that time series. So we actually have stickleback bones at different time points, and we can see how these allele frequencies are actually changing. So I'll just quickly check my time. Right. Gosh, I need to go a bit faster, but this second case study is going to be looking at ancient killer whale genome, and how just a single representative ancient genome can give us some signatures of the sort of, give us some resolution of the temporal pattern of changes in the allele frequencies, in this case associated with purifying selection. So killer whales are actually one of the most widely distributed mammals. So I think the closest rival is us and the brown rat. They're found from the Arctic to the Antarctic and everywhere in between, but they're at the highest densities in high latitudes. So these are really productive regions. And obviously, the higher the productivity, the more food there is for these top marine predators. Now, what you can see here is actually two habitat maps with the ocean coloured by the relative habitat suitability. The turquoise colour is the extent of the permanent sea ice, and that's the summer distribution, I think. No, that's the winter distribution. So the top is during the habitat prediction during the last glacial maximum, and the bottom is during the present day. And we can see actually that the core suitable habitat is also these regions in high latitudes. So what's surprising, in fact, is that the effective population size is highest in actually low latitudes. And a lot of these high latitude populations, if I take Norway as an example, we can see that it's got quite low effective population size. But we have literally hundreds of killer whales off of the coast of Norway, feeding on a huge resource of the Norwegian spring spawning herring. Whereas if you go to somewhere like, for example, the Maldives, you can see, in fact, the highest effective population size was found in sample from the Maldives. So we actually use FITA and extrapolate up. Then, I would tell you that the chance of you being attacked by a killer whale is quite low, because they just really are not there in any great numbers in the Maldives or in the Indian Ocean at all. So it's quite surprising to see that. Now, we also see that we talked about runs of homozygosity earlier with the stickleback. Here we're looking at runs of homozygosity in these killer whales, and we see the same pattern here. And what we see in here is different lengths of these runs of homozygosity. So they've been put in length categories. So here it's 300,000 base pairs to 500,000 base pairs, and so on. And why we're putting them in these bins is it gives us a little bit of an idea of when the inbreeding happened. So essentially, when the two haplotypes, identical haplotypes, were shared, and the further back in time, the more recombination events that could have occurred since. And so we tend to see like these smaller ROH being associated with time, well, several, based on our estimates with recombination rate, probably mid-early Holocene events. And we see the same pattern in our high latitude population. So here we've got, for example, a Norway killer whale in black, a North Pacific killer whale from Alaska in blue, and an Antarctic killer whale in red. And we can see all of them share this excess of quite short ROH, which we associate with a colonization, a founder effect, a little bit like we were seeing in our stickleback fish, going back in during the sort of early Holocene, probably when that habitat became available for the first time after the retreat of the ice sheets again. And here we can see three representative low latitude populations, and we can actually see quite a low number of ROH. And what we think is happening here is that these populations with very different genetic backgrounds are just very occasionally intermixing And then you get this recombination between these very different genetic backgrounds, and it introduces heterozygosity into the population. And you end up with something essentially the sort of natural equivalent of genetic rescue. And we see a little bit of that here. So here we've got like an Icelandic killer whale. We've got a marine mammal-eating killer whale from Alaska, which doesn't show this same signature. And there's actually a good reason for that. It seems like these transient killer whales belong to an earlier colonization event from an earlier glacial period. And we have two F1 Iceland transient killer whales from captivity. And we can see that they have much lower numbers of runs of homozygosity with these mid-lengths than we see in the Icelandic killer whale. So we can see that those runs of homozygosity are being broken up. So this is my visualization of how killer whale population structure looks worldwide. Essentially, that there is these diverse populations sporadically interbreeding with one another, and so we end up with the variation being essentially shared more or less globally. It might not be in complete equilibrium between populations, but it means that a lot of the variation is there, a standing variation in the pool. Again, just like it was in sticklebacks. And then at the range margins, we see the influence of these serial founder effects. So essentially do a step-by-step colonization after the retreat of the ice sheets and the availability of new habitat emerging, or perhaps just a continuous range expansion. But in each case, we're seeing a drop of effective population size and also diversity at the range edge, at the leading edge. And we're going to expect from that there to be reduced effective population size, reduced efficacy of selection, and that would be both on adaptive traits, but also on purifying selection. So essentially there's deleterious mutations which are harbored perhaps neutrally in heterozygote genotypes in this gene pool in tropical low latitude waters. Those could rise to high frequency and be exposed in these homozygous genotypes and these ROH at the range margins. So to try and look at this and to compare the differences in efficacy of selection between populations using just a single sample, which is what we've got really with our global data set, we're using this RXY statistic, which was conjured up by David Reich's group, which is a Doet-Oll paper in Nature Genetics, looking in humans and also comparing humans to Neanderthals. And then also in mountain gorillas in Zoo et al. In Science from a few years ago. And it's based on the assumption that if you count the numbers of synonymous mutations and non-synonymous mutations that have occurred in one population, but which are not shared with another population, they should be roughly equal to those that are in population Y, but not found in population X. And this is assuming that there's no interpopulation variation in the mutation rate, and also under neutrality. Well, if there's differences in effective population size and the efficacy of selection, however, we may see an accumulation of deleterious mutations in one population compared to the other. And actually what we see, the most striking result, is in Iceland and Norway. And actually the Maldives shows kind of the opposite signature. And so what we're seeing here is RXY non-synonymous is the counts of synonymous alleles that are in, the counts of non-synonymous alleles divided by the counts of synonymous alleles that are in Iceland and compared to all of these populations on the y-axis, or in Norway compared to all these populations on the y-axis. And what we see is a deficit of non-synonymous mutations compared to these populations in the Iceland and the Norway sample. And it's significant when we use like a weighted jackknife approach to generate a Z score. So so basically it's more than 4 standard deviations from 1 in each case. And same with the Maldives, this has a significant excess of non-synonymous mutations compared to what would be expected from compared to what's found in these other populations. Now, what we were interested in was trying to find the timing of this. Is this related to this colonization process? And perhaps when these, I guess I should say as well, there's an assumption here, which is that non-synonymous mutations are going to be deleterious, or very weakly deleterious perhaps, and synonymous mutations are neutral. Now that is an assumption that's likely not true in every case, but broadly speaking, it's probably going to be representative. Now, by having an ancient sample, we hope to try and pinpoint a little bit when this, what seems to be purifying selection and purging of non-synonymous mutations in the Norway and Iceland sample happened. And to do this, we sequenced the genome of a killer whale. And this was from the collections in Copenhagen Museum, and it was dated to close to 8,000 years ago. So it's these teeth here from this picture, and you can see here we've got individuals which are also from Denmark. So this was from Denmark, and here are individuals also from Denmark, and you can see that all three of them have got these worn down teeth, which are worn down actually to the gum line. Now here's a photo I actually found online, and you can see really nicely how these worn down teeth look. And this is the Norwegian killer whale feeding on herring. And what they do is just suck these herring up one at a time. And we believe it's that suction action that actually wears down the front teeth. And you can also see from the stable isotope values that there's a little bit of, well, I would say quite a big difference in the trophic level that each of these animals is feeding upon these museum samples. Now these two are historic, I would say, like around roughly 150 years old each, whereas this one here is truly ancient and is the 8,000-year-old one. We can see that this animal here was feeding at a much higher trophic level, and actually this animal here in the Copenhagen collection is actually referred to in Jules Verne's 20,000 Leagues Under the Sea. Says, I can believe a certain gentleman from Copenhagen who claims to have taken 13 porpoise and 15 seals from the stomach of one dolphin. It is true, an orca belonging to the biggest known species that sometimes exceeds 24 feet. So the gentleman that he's talking about is Eschrich, who was the curator of the Natural History Museum in Copenhagen at the time, and who documented the necropsy on this killer whale, where he just pulls these seals and porpoises from the stomach, and the seals actually still had the pelts attached to the heads. So it seemed that the killer whale was whacking them with the tail to split them open, which is exactly how we've seen lone male killer whales feed on harbour seals around Shetland. And this actually fits in with, so I guess, sorry, just to explain as well, although we're seeing this difference in trophic level, we're seeing this similarity in the tooth wear. So it suggests that these killer whales are feeding on the same thing for part of the year, but perhaps changing their diet sporadically or seasonally. This one feeding at a higher trophic level, and perhaps this one continuing to feed on herring year round. And it looks like our ancient sample was feeding at a high trophic level. And this is exactly what we find from the fieldwork as well. So this is us up Shetland, this is me driving the rib. And this animal here is the Icelandic killer whale whose genome we sequenced and is included in this study. And this animal feeds on herring in the winter off around Iceland. And while the rest of that population, well, a lot of the rest of that population follow the herring to spawning grounds, this killer whale and the group that it belongs to, plus several other groups, move over to Shetland for the summer, and there they're mostly feeding upon seals. So you get this idea of how the stable isotopes can tell you one thing, and the tooth wear tells you another thing. Now, that was just key to give you a bit of background that the ancient sample seems to have been ecologically quite similar to the Iceland and Norway sample. It was geographically quite proximate. It was kind of an intermediate place found off of Denmark. And we can see that genetically, it seems to sort of be the closest along this PCA from the more low latitude population samples here. Now we do a number of other different tests as well to try and get a feel for how directly ancestral this ancient sample was. And it seems like it's not completely directly ancestral, but it's very close to. In other words, when you go back to the true ancestor that's shared between that ancient sample and the Norway and Iceland one, there's very little drift along to the ancient sample from that recent common ancestor. Now, the ancient sample also shows these characteristic damage patterns. So when we're working with ancient DNA, we typically get a DNA sequence that's different from the true DNA sequence, because different chemical processes change that sequence through time. So deamination, for example, changes cytosine to uracil. And the sequencer can't read uracil, so it interprets that as thymine. So we get this excess of C to T changes. We can see here big proportion of T's where we expect a C from based on the reference. And we see the reverse complement of that at the 3' end. So we see these G's to A's. So because of that, we have to exclude all transitions from the analysis, and we just work with transversions. And we do a normalization based on the synonymous changes, because of course the branch to the ancient population is going to be shorter. And I must hurry up because I'm getting quite close to the end of my time. But the key take home here is that essentially we see a similar pattern in this ancient sample to what we see in Iceland and Norway. So we do see this excess of synonymous to non-synonymous alleles, and but it's basically the Norway and Iceland samples are much more seem to have purged much more of the non-synonymous mutation load. So the ancient sample is intermediate, and it suggests that this purging is something that takes place over thousands of years. So this idea, sort of quite a theoretical based idea, that exposing these potentially recessive, likely deleterious recessive mutations in homozygous genotypes might expose them to selection, and we can see that process might bias, and we compare to some additional modern killer whales, and we see the same results. We also see, so here we've got, we're just looking at alleles which are in the derived non-synonymous state in the ancient sample, and we can see that they are shared globally, maybe not amongst every individual, but we can see that really what we're looking at is standing variation that's present in the global population data set. And the non-synonymous mutations that were purged and were missing from the Norwegian killer whale genomes, they were found globally shared, as global shared variation in heterozygote genotypes, significantly more than expected under Hardy-Weinberg equilibrium. So in other words, it's consistent with this idea that they're potentially recessive in nature. And so in the homozygous genotype, perhaps selection is more likely to act upon them. And all of this fits with our theoretical model that a lot of this variation is present in the global population at low latitudes, and it's really when it comes to the colonization of high latitude areas, they go through this colonization process, and that's when the selection processes really start to kick in, when a lot of this recessive variation is exposed to selection. So just to conclude, even single genomes can increase the resolution of our understanding at the timing of selection at the genotype level. So, you know, if you think, oh, you know, what can I really do with one sample? You might go to the museum collections there and see something and you think that's really interesting, but it's just one sample. Well, you never know. These ancient samples nearly always catch us out, and they tell us something quite different to what we potentially would have assumed just based on the modern samples that we have. And they give us this temporal context to the evolutionary processes that we just can't get from modern samples. The Neanderthal genome is probably the best example. You know, it's one we had for a long time, one good sample, the Altai Neanderthal genome, and we've learned so much about our own evolutionary history from that. So definitely don't be discouraged. You've got to start somewhere, and hopefully I'm going to build up to what Ludovic Orlando has done with with horses, with killer whales and sticklebacks. But it's good to have some nice insights along the way, even with small samples. So I will stop sharing my screen. Thanks so much for listening.